Hello students, welcome in Babita Science tutorial. Let's continue our chapter The Fundamental Unit of a Life. In this video, I will explain you activity 5.3 and this activity is based on the osmosis process. Let's start now. Here 2 points is given. First is your this one and second one is this one, but both are depends upon the osmosis process. In this one you will take egg, okay, and dissolve this egg in the hydrochloric acid. When we dissolve in the hydrochloric acid, then you will easily remove its outer shell, and its outer shell is made up of the calcium carbonate. Okay, now you have the egg only in which very thin layer is present. Now see student, here take the one beaker, okay, and in this beaker you will put the water in it, okay, and then after you will keep the egg in this water. Then what happened, keep this egg in this water for 5 minutes, and after 5 minutes you will see that the egg becomes a swell up. Why this egg become a swell up? Because the water will goes inside it, okay. When the water will goes inside it, then it will increases the size of the egg, and this is the example of a hypertonic solution. In the hypertonic solution, the water concentration is a maximum maximum as compared to the cell of a egg. You will see here the percentage of the water is very less in the egg as compared to the percentage of water from outside the cell. So this is the example of the hypertonic solution, and here osmosis process takes place. But in the second case, same thing you will, when you will put a same egg in the hypotonic solution, but this hypotonic solution is a salt solution. Means in the second part, when we keep this egg in the salt solution, okay, then what happened here, this egg shell start to shrink. Why this egg is start to shrink? Because salt solution is a more concentrated, okay. Then what happened, the egg which is present inside this water, okay, it will start to exoosmosis. Exoosmosis means the water is comes out from this egg outside, okay. That process is called the exoosmosis. And in the first one what happened, water is comes inside the eggshell. That process is called the endo osmosis, okay. Endo, this one is the endo osmosis, and this one is called the exo osmosis. When the water is comes out from the eggshell, okay, that process is called the exo, and when the water is goes inside the eggshell, that process is called the endo osmosis. Then you will see here osmosis process takes place in the both, but difference is that in the first one we have taken the pure water only. That's why the swells, the egg becomes a swells because water is passes into it by osmosis. And in the second cases, we have put the same egg in the salt solution, okay, but what happened here, salt solution is a more concentrated. That's why the water which is present in the egg cell is comes out, and that's why this process is called the exoosmosis. Now let's proceed to the another activity. Here is the 5.4 activity. In this activity, they want to say put dry raisins or apricots in a plain water and leave them for some time. Okay, then passes them into the concentrated solution of sugar or salt. You will observe the following. Same thing, student, here same case, okay, but materials we have taken different. Here we take the dried raisins, okay, and apricots. First we will take the beaker, and in this beaker water is present, and put a resins here, okay, and this one is your pure water, okay. Now what happened, the resins becomes a swell up. because here water is pure here, and this water is goes inside this resins, okay, then it will increase the size of the resins, okay. Then again here is a hypertonic solution. Percentage of the water is more as compared to the percentage of water in the resin. Then here is a hypertonic solution, and osmosis process takes place. But students, when we place same thing, okay, in the suppose that if we will take this one is a bigger, but when we take the salt solution here, okay, and put the resins here, okay, then what happened here, the resin is start to shrink. Why this resin just start to shrink? Because the salt is more concentrated, and the water which is present inside this resin is comes out, okay, and this resin becomes a shrink. You will see here each gains water and swell up when placed in water, okay, and in the second case, when they placed in the concentrated solution, it loses water and consequently shrink. Now in the very simple way, I want to explain you that when you put anything in the simple water, it will be swell up because water is a plain, okay, and it will goes inside the substances and it helps in the swelling. But when you put in the salt solution, then what happened, exo-osmosis process takes place. Water is that comes out from the substances, and in this way the substance or the shell becomes a shrink. I will give you one example more in the daily life. Let's take the example of a mango pickles. Okay, when we add the salt in the raw mango, when we cut the mango and you will add the turmeric and salt, then what happened? What is the comes out? No, then here again exo-osmosis process takes place, and the shell become the shrink. Shell means shell of a mango becomes a shrink. Let's proceed to another paragraph here. Now in this paragraph here, unicellular freshwater organisms and most plant cells tends to gain water through osmosis. Yes, true. Now unicellular example you will take the amoeba. Okay, amoeba can take the water with the help of the osmosis process, and the plant cells, suppose this is the plant cells and it is the roots, okay, and the plant's roots can also absorb the water with the help of the osmosis process. You will see here absorption of water, plants roots, it also an example of a osmosis. Then students here sometimes examples are also asking about the osmosis. Give the example of a osmosis, then you will give here give the example of a amoeba and the plant roots, okay. Then let's proceed to another paragraph here. Now thus diffusion is important in exchange of gases and water in the life of a cell. Very true. Diffusion and osmosis process helps in the life of a cell. Diffusion helps in the exchange of gases and water. Osmosis helps in the water, okay. Then in addition to this, the cells also obtain nutrients from its environment. Different molecules move in and out of the cells through a type of transport requiring use of energy. Student cells, in the cells gases comes out, and water also goes out and comes in due to the process of a diffusion and osmosis process. But some molecules also comes in and goes out through a types of transport, okay, through a types of transport which can needs the energy also. We can see that with the help of this transport, the molecules moving and out of the cells, okay. Now let's proceed to another paragraph here. Now the plasma membrane is a flexible and is made of the organic molecules called lipids and proteins. I already give you about the components of the plasma membrane, that is the lipids and proteins. Okay, and these lipids and proteins makes the plasma membrane flexible. Okay, however, we can observe the structure of the plasma membrane only through an electron. With the help of the electron microscope, we can easily see the structure of the plasma membrane. It's not possible to see the structure of the plasma membrane with the help of the compound microscope, but for the with the help of electron microscope, you can easily see the plasma membrane. Okay, it's a lot of difference between the compound microscope and the electron microscope. Now, in another paragraph, the flexibility of the cell membrane also enables the cell to engulf in the food and other material from its external environment. Such process is called the endocytosis. Amoeba acquires the food through such process. Students, here endocytosis is a very important, and this process can takes place in the amoeba. Okay, now see here, I told you dear that the cell membrane is a flexible. Okay, now see here, because this always question asking in the exam, which process takes place in the amoeba. Student, this is the structure of the amoeba. Okay, and the cell membrane of the amoeba is a flexible. Okay, when any food material is comes inside this amoeba. Okay, and this its cell membrane is a very flexible. Okay, then when any food material is comes inside this cell membrane of the amoeba, that process is called the endo means inside, cyto means cells. Okay, the endocytosis, when any material comes inside the cell, that process is called the endocytosis, and this process can take place in the amoeba. So that is a very important definition. What is the endocytosis, and and which process takes place in the amoeba. So once again, you will listen the definition of the endocytosis. Endocytosis is the ingestion of materials by the cells through the plasma membrane. Okay, and it can take place in the amoeba. Student, got it here? What is the meaning of the endocytosis? Because this one is a very important definition and asked to you in the exam time also. Now, in activity 5.5 is nothing. They want to say find out about electron microscope from the resources in the school library or through the internet and discuss it with your teacher. You can see the structure of the electron microscope with the help of the net. Okay, and you can easily find it this one in the Google also. Now here some questions are given. You can try this question, and I will explain you next topic cell wall in my next video. Till then you can understand this one all. This activity 5.4, 5.3, and here hypertonic solution. What is hypertonic solution? Isotonic solutions and hypertonic solution. Okay, and endocytosis is given, very important. Plasma membrane components is given. Okay, example of the osmosis is asking in the exam time. So this one is the most important. You can focus on it and you can learn it. If you have any problem, comment me in my comment box. Till then, bye and have a nice day.